just for a few minutes because everything I am adding here as a link.
Okay.
Yes, I have clearly mentioned that this link is coming from GEO.
Yeah.
Okay,
Yeah.
so there will be no issues
Mm.
because here it is just click here. I
Mm.
want to show here, but today you just need to read the things. Okay, and you also need to download this paper,
Mm.
this one,
Mm.
which they have referenced here.
Mm.
Okay,
Mm.
when you will download this paper, then you have to read it properly. Okay, but
Yeah.
I'm still trying to paste it on this uh,
PPT.
PPT. So everything will be here. There is no need to close anything. Okay.
Okay.
Yes. It is just a matter of time. That's it.
Yeah. Yeah.
Yes. So this is already here now. It's a complete detail.
Yeah.
Okay. So there is nothing that we need to bother much. It's a very simple way. Everything is here. This one is the link for the references here.
Yeah.
And you just download this and the details you will find on the website for which I have given the link. In
Mm.
case if you are not able to open this link,
Mm.
Google this database,
Yeah.
you will get it. This is the GData ID, this one.
Okay.
Yes. So perfectly, this is all right.
Mm.
Now your task will be just to go for here. I'm just showing you
Yeah.
because there are three different data types, three, four. So
Mm.
you need to group them together.
Okay.
Okay. So you have to just go define group.
Mm.
In defining group, you will be getting, for example, I wrote simple BM. That means bone marrow,
Mm.
nothing else. Okay.
Mm.
And then second group will be bone marrow with something selected cells or something. CD34 positive cells.
Mm.
So you can write BM CD34. That's it. Okay.
Yeah.
And when you write this, then you just click on all the BM samples together.
Mm.
Let's say here, it is selected yellow color. Just you
Mm.
need to look for the yellow colors.
Okay.
Where it is written, one type of sample will be going to BM, bone
Mm.
marrow.
Mm.
Then another sample will go to PMCD34. You just look for this and click and then click here. Select, click.
Mm.
For example, I selected this one, click
Mm.
here. Then again, then like this. Now it is selected. Again, do like this. You can do here one by one.
Mm.
There are not many samples, so you can do like this.
Okay.
Another group is leukemia, but you can go later on.
Okay.
You have to compare two groups in one time.
Okay.
You cannot compare in one time more than two groups.
Mm.
Is it clear now?
Yeah.
So first you are comparing direct bone marrow samples with bone marrow samples where CD34 positive cells were selected. And then go for differential expression analysis between these two cells. Click here, analyze.
Okay.
After few seconds, you will be getting the result on this screen.
Mm.
You are not using any software for differentially expressed genes. You got it here?
Yeah.
All the data is here now.
Yeah.
What you need to do here? So you just go for downloading the things. Okay.
Okay.
Now, how will you download it here? This is download full table.
Mm.
If you will click, you will get a text file.
Mm.
All the list of the genes. You should not copy from here.
Mm.
You direct click here, download full table.
Mm.
Once it is done, and this is also the differentially expressed genes list.
Okay.
Bone marrow versus bone marrow CD334 positive cells.
Yeah.
You got this thing? Red are
Yeah.
upregulated. This cyan color is downregulated genes.
Okay.
Black is insignificant.
Okay.
And it is clearly mentioned here.
Okay.
But I will explain you how will you understand which genes are up or down regulated.
Hmm.
So the first stage of learning for today in this sense is now something that is called gene expression. Are you able to see?
Yeah. Yeah.
Profiling. I make it bold, sorry. Okay.
Yeah.
In gene expression profiling, there are few things which you calculate. First thing, p value. Second thing, fold changes. These are the two parameters which you need in order to say that this gene is up or down regulated gene.
Okay.
And so we say differentially expressed genes. And in shortcut, we write DEGS.
Okay.
Now I think it is very, very clear.
Yeah. Of course.
Yes.
This
So,
this P value we use for statistical analysis, is it the same or no? It's
yes,
okay.
this is p value is coming from your statistical analysis.
Yeah.
So now you are basically, I'm showing you how will you make your workflow also.
Hmm.
In gene expression profiling, you select the data set.
Hmm.
Okay. This is the first thing.
Hmm.
Then selection of data set,
Hmm.
which you have already selected.
Yeah.
Then calculation of p-values and fold change.
Okay.
Next part is after calculating fold change and p-values, what you do? Assign a threshold. Assign the threshold level for considering the genes as differentially expressed.
Yeah.
And that will be p-values must be less than equal to 0.05.
Yeah.
Fold change must be greater than equal to something. Let's say here I'm saying you can consider for cancer plus 2.0 for upregulated genes. Okay.
Hmm.
And for downregulated genes, it must be less than equal to minus 2.0. Okay.
Yeah.
So you always need to keep, if you are keeping for upregulated plus 2, downregulated must be minus 2 also.
Hmm. Okay.
Yes. So both the values must be same except in terms of plus minus sign.
Okay.
And p value will be. So it's not and or. P values and fold change, both the things should be in the limit of threshold value.
Threshold. Yeah.
Is it now clear?
Yeah.
So here you write down regulated.
Down. Yeah.
Very clear things here.
Yeah.
Okay. So now I highlight is as red and this one as, let's say, blue.
Hmm.
And this will be for both the cases. So keep it as something dark green.
Okay.
That means These three colors have different meanings here.
Okay.
So do you get the things?
Yeah.
Okay. Now, what is the next part after you do it?
But my question is, where I will do it? Like, is there any tool or how I will analyze it?
Yes, that is why I'm coming.
Yeah. Okay.
Yes.
Mm.
So this, when you will download this file,
Yeah.
you will get all the adjacent p-values or adjusted p-values. P-values, log fc is the fold change,
Mm.
and this is gname. So after downloading the table, you copy them and paste it into Excel file.
Mm.
Delete all the columns except log fc, gene name, p-values.
P-value. Okay.
Delete everything.
I have to keep the ID as well, right?
id, gene symbol,
Mm.
log fc,
Mm. And
p-value.
P-value.
That's it.
Okay.
There is no need of this id. Is it clear?
Yeah.
So your question was from where you will get, you already have it here.
Okay.
Why I am showing this? This is first step of your understanding. The rest you can do it by when you will be running MATLAB in
Mm.
MATLAB on your own.
Mm-hmm.
How will you do it? I will also explain it. But
Okay.
instead of directly jumping there, because you are still in basics of MATLAB coding,
Of
so
course.
you should not directly go for coding. You should start from one way, calculating the learning bioinformatics also, and in parallel, you should try to practice some basics in MATLAB also.
Mm.
Is it clear now?
Yeah.
So this is overall what I was trying to say it for today. Okay.
Okay.
And I think this will be enough to keep you busy the entire week from Monday to Friday.
Okay.
Like MATLAB practice, some bioinformatics tasks, you have it already, and then you will have more things. So what you will be doing in Excel file, you will need to play more and more in Excel file,
Mm.
okay,
Okay.
where you will be sorting the genes based on fold change
Mm.
from higher values to lower values.
Okay.
So from top, all the values, it will be for upregulated genes, and from the bottom, most of the things will be for downregulated genes.
Okay.
That's it.
So if I find something like out of the range that you set, should I delete them or should I put them?
No, you should keep it into one sheet, everything, all the values.
Mm-hmm.
In the same Excel sheet, you file, you create another sheet
Mm-hmm.
where you will have just anything above plus 2.0
Yeah.
and anything below minus 2.0. The middle genes will be deleted.
Okay.
So one will be clean file with only up and down regulated. So
Okay.
you need to create four sheet. First sheet in Excel file will be with all the values,
Okay.
where you will have all the gene names, log
Mm.
FC and
Mm.
p values.
Mm.
Then you create another sheet that is for upregulated genes, all the genes with more than plus two values,
Mm.
and followed by their p values and gene names.
Mm.
Third sheet will be with all the genes which have fold change less than equal to minus 2.0, followed by their p values and gene names.
Yeah.
Then fourth sheet will be combine all the up and down regulated genes in
Okay.
one file. That's it. Is it clear for understanding?
Yes.
So this is overall for today.
Okay.
I will send you few more things today, and that thing will be for MATLAB basic practice.
Okay.
Yes.